over development of the brain, module 4.2. Okay, so think of all the things you could do that you couldn't have done a few years ago. Okay, so analyzing statistics, reading a foreign language, writing brilliant critiques of complex issues, and so on. If you develop these new skills because of brain growth, many of your dendrites have grown, new branches, but your brain as a whole hasn't grown. Now think of all the things that a 1-year-old can do. Have they developed their new skills because of brain growth? To a large extent, yes, although the results depend on experiences as well. In this module, we'll consider how neurons develop, how their axons connect, and how experience modifies development. So, maturation of the vertebrate brain is a big topic. The earliest stages of development are remarkable, and they're similar across species. A series of genes known as the homeobox gene is found in vertebrates, insects, plants, even fungi and yeast. Regulate the expression of other genes and control the start of anatomic development, including such matters as which end is the front and which is the rear. All these genes share a large sequence of DNA bases. A mutation in one homeobox gene causes insects to form legs where their antennae should be, or to form an extra set of wings. In humans, mutations in homeobox genes have been linked to many brain disorders, including mental retardation and physical deformalities. The human central nervous system begins to form when the embryo in a 2-week-old. The dorsal surface thickens, and then large, long, thin lips rise, curl, and merge, forming a neural tube that surrounds a fluid-filled cavity. As the tube sinks under the surface of the skin, the forward end enlarges and differentiates. Then, the hindbrain, the midbrain, and the forebrain. The rest becomes the spinal cord. The fluid-filled cavity within the neural tube becomes the central canal of the spinal cord, and the 4 ventricles of the brain containing the cerebral spinal fluid. The first muscle movements start at age 7.5 weeks old, and their only accomplishment is to stretch the muscles. At that age, spontaneous activity in the spinal cord drives all the muscle movements, as the sensory organs are not yet functional. That is contrary to what we might guess. We start making movements before we start receiving sensations. At birth, the average human brain weighs about 350 grams. By the end of the first year, it weighs 1,000 grams, close to the adult weight of 1,200 grams to 1,400 grams. In early infancy, the primary sensory area of the cortex, responsible for registering vision and hearing, and other senses, are more mature than the rest of the cortex. So you got the forebrain, the midbrain, and the hindbrain. And then that develops, and then it starts the cranial nerves, the spinal cord, and then it becomes, the forebrain gets big, the midbrain becomes this little piece in the middle, and the hindbrain and the spinal cord are kind of like gapped out. Eventually, when it's after 11 weeks, and you're actually having the baby, the forebrain will be developed. The midbrain will be hidden under the forebrain. The cerebellum will be at the back, where the hindbrain originally is, and then the medulla sits right under the cerebellum. So the growth and development of neurons. Neuroscientists distinguish several stages in the development of neurons. There is proliferation, is the production of new cells. Early in development, the cells lining the ventricles of the brain divide. Some cells remain where they are, as stem cells, continuing to divide, whereas others migrate to other parts of the nervous system. In humans, Most of the migration occurs before birth, but a small number continue to migrate for the first few months after birth. Early in development, the primitive cells are not yet identifiable as neurons or glia. They begin to migrate, the neurons and the glia, and chemicals known as immunoglobulins or chemokines guide neuron migration. A deficit in these chemicals lead to impaired migration or decreased brain size and mental retardation. As a cell differentiates into a neuron, it begins to form its dendrites, axons, and synapses. Synaptogenesis, the formation of synapses, begin long before birth, but it continues throughout life. As neurons form new synapses, they discard the old ones. This process generally shows in older people, as does the formation of new dendritic branches. A later and slower stage of neural development is myelination. Myelination. It's like myelin sheath, but myelination. The process by which glia produce the insulating fatty sheaths that accelerate transmission in many vertebrate axons. Myelin forms first in the spinal cord, and then in the hindbrain, midbrain, and forebrain. Myelination continues gradually for decades and increases as a result of learning a new motor skill. Okay, so there's a lot of stuff about frogs and lizards, and I kind of want to know more about Darwinism. So these results suggest a general principle called neural Darwinism. In the development of the nervous system, we start with more neurons and synapses than we can keep. And then a selection process keeps some of the synapses and rejects others. The most successful combinations survive, and the others fail. The principle of competition is an important one, though. Although we should use the analogy with Darwinism, evolution continuously. Mutations in the genes are random events, but neurotrophins steer new axonal branches and synapses in approximately the right direction. Initially, the sympathetic nervous system forms far more neurons than it needs. When one of its neurons forms a synapse into a muscle, that muscle delivers a protein called nerve growth factor, or NGF. That promotes the survival and growth of the axon. An axon that does not need or receive NGF disintegrates, and its body dies, and each neuron starts life with a suicide program. If its axon does not make contact with an appropriate postsynaptic cell by a certain age, the neuron kills itself through a process called apoptosis. It's a programmed mechanism of cell death, an injury, or a toxic substance. NGF cancels the program for apoptosis. It is the postsynaptic cell's way of telling the incoming axon, "I'll be your partner, don't kill yourself." The sympathetic nervous system's way of overproducing neurons and then applying apoptosis enables the CNS to match the number of axons to the number of receiving cells. Of course, there's going to be cell loss during development. The number of motor neurons in the spinal cord of human fetuses is highest at 11 weeks and drops steadily until about 25 weeks. Axons that fail to make synapses die. Nerve growth factor is a neurotrophin, meaning a chemical that promotes the survival and activity of neurons. In addition to NGF, the nervous system responds to brain-derived neurotrophic factor, or BDNF, and several other neurotrophins. Neurotrophins are essential for growth of axons and dendrites. There's a vulnerability at developing the brain. Don't drink when you're pregnant, okay? Because the infant brain is highly vulnerable to damage by alcohol. Children of mothers who drink heavily during pregnancy are are born with fetal alcohol syndrome, a condition marked by hyperactivity, impulsiveness, difficulty maintaining attention, varying degrees of mental retardation, and motor problems, heart defects, and facial abnormalities. Drinking during pregnancy leads to thinning of the cerebral cortex that persists to adulthood. More drinking causes greater deficits, but even moderate drinking produces a measurable effect. Educators have long operated on the assumption that training children to do something difficult will enhance their intellect. In general, long ago, British schools taught children Greek and Latin. Today, it might be calculus, but in either case, the idea is not to teach something challenging and hope children get smarter in other ways too. The psychological term is far transfer. Near transfer occurs if training on one task produces improvement on a similar task. In general, far transfer is a weak effect. Many studies have basically attempted to improve memory or intelligence by computerized tasks that practice verbal and spatial skills. Despite high claims by the publishers of these programs, most studies show little or no improvement in real-world performance. Suck on that, intelligence apps. So another part of everything would be when a brain recognizes it's gone too far, or when brain's reorganization goes too far. In some cases, the later process does occur, such that stimulation on one finger excites mostly the same cortical areas on another finger. If you cannot clearly feel the differences between one finger and another, it is difficult to move them independently. Furthermore, the motor cortex changes also. Representation of the middle finger expands, overlapping and displacing representation of the index finger and little finger. One or more fingers may go into constant contraction. Moving one finger without moving another becomes more difficult. This condition is known as musician's cramp, or more formally as focal hand dystonia. This can threaten a musician's career. Similar problems occur in writers, surgeons, golfers, or anyone else who repetitively practices precise hand movements. So, in summary, in vertebrate embryos, the central nervous system begins as a tube surrounding a fluid-filled cavity. Developing neurons proliferate, migrate, and differentiate, and develop synapses and myelin. Neuron proliferation varies among species mainly by the number of cell divisions. Migration depends on chemicals that guide immature neurons to their destinations. In adult vertebrates, new neurons form only in a few parts of the brain. Adult humans form new olfactory receptors, but the brain develops new neurons only in the hippocampus and the basal ganglia, both of which are important for new learning. Growing axons find their own way to the right locations and then arrange themselves over a target area by following chemical gradients. After axons reach their targets based on chemical gradients, the postsynaptic cell adjusts the connections based on experience, accepting certain combination of axons and rejecting others. This kind of competition among axons continues throughout life. Initially, the nervous system develops more neurons than will actually survive. Axons of the sympathetic nervous system survive only if they reach a target cell that releases to them nerve growth factor. Otherwise, they die in a process called apoptosis. Apoptosis also occurs in the brain, but the factors controlling it are less well understood. Prenatal exposure to alcohol increases apoptosis. The developing brain is vulnerable to chemical insult. Many chemicals that produce only mild, temporary problems for adults can impair early brain development. At an early stage of development, the cortex is sufficiently plastic that visual input can cause what would have been the auditory cortex to develop different properties and now respond visually. Enriched experience leads to greater branching of axons and dendrites, partly because animals in enriched environments are more active than those in deprived environments. Specialized experiences can alter brain development, especially early in life. For example, in people who are born blind, representation of touch and hearing expands in these areas, usually reserved for vision. Extensive practice of a skill expands the brain's representation of sensory and motor information relevant to the skill. For example, the representation of fingers expand in people who regularly practice musical instruments. Although controversy it remains, several studies report that even brief practice of a skill such as juggling can produce measurable changes in brain anatomy. Although expanded representation in the brain is ordinarily a good thing, it can be harmful if carried too far. Some musicians and others who use their hands for many hours each day develop brain changes that interfere with their ability to feel or use one finger independently of the others. Compared to adults, adolescents tend to be impulsive and centered more on present pleasures than future. In most cases, risky behaviors in adolescence probably reflect increased drive for excitement, more than lack of ability or inhibitive functions or impulses. On average, people in old age show declining memory and reasoning and shrinkage of certain brain areas. However, these areas do not apply to all individuals or all situations. On average, physically fit people tend to maintain their cognitive abilities. Many older people compensate for their inefficiency of certain brain functions by recruiting activity in additional brain areas. Thank you for listening. Hit subscribe if you want to know more about biological psychology. Thank you.